Good evening and welcome to this event, which is part of our Transforming Healthcare series. You're gonna hear tonight about unlocking the potential of cellular therapy. I'm John Riley, the Vice Chancellor for Health Affairs and the Dean of the School of Medicine here in the Anschutz campus. And on the behalf of Chancellor Elliman and myself, I'm happy to welcome all of you here tonight. This program, series of programs, started 7 years ago as a way to allow interested people to learn about some of the exciting science and developments that were going on this campus. And then we had this little sort of COVID detour 3 years ago where we repurposed it to talk about the pandemic, but thankfully we're now back on track. And what you're going to hear tonight, I think, is some really exciting work that's done by multiple faculty members on our campus and has already directly affected the lives of many patients, one of whom you'll hear from tonight, and has the potential to affect many more. Now, we chose this theme tonight for two reasons. One is because it is a very hot and exciting area in translational medicine research and therapeutics, particularly cancer therapeutics. And it's a way to celebrate a milestone for this campus and the University of Colorado, which is the formal establishment of the Gates Institute. And so I want to say a word about, before we get into the science and I introduce our speakers tonight, I want to say a word about the history of the Gates Institute. 17 years ago, it was the vision of Charles Gates, an entrepreneur here in Colorado, who became very attracted by the potential of regenerative medicine to start a research program to try to accelerate the movement of fundamental scientific discoveries into meaningful therapy for patients. And so the first name for this program was the Gates Regenerative Medicine Program, and a few years later it became the Gates Center. Many of you know Charles Gates was a well-renowned entrepreneur here in Colorado. Had a number of sayings that he used to encourage entrepreneurship and inculcated those values in his two children, Diane and John. One of my first events here, actually my first formal event here as Dean at the beginning of April in 2015, I'd like to tell everybody I started on April Fool's Day, was actually the dedication of another milestone for this campus, which was the opening of the Gates Biomanufacturing Facility. So early on, the Gates program was fortunate to recruit a visionary leader in Dennis Roop, a renowned scientist who was able to attract other scientific talent to join him. And the missing link between the lab and patients was a facility that could manufacture the cells and proteins that would be infused into patients to produce a therapeutic effect. And the Gates Biomanufacturing Facility filled that missing link. And today, it's producing therapies that are in active clinical trials on this campus and at other sites across the country. It's really that facility and the program that was the vision of the Gates family has really enabled this campus to be at the forefront of the science that you're going to hear about tonight. Along the way, the Gates Foundation, and particularly Diane Wallach Gates and her late brother John, have been essential partners, both in terms of the support they've provided us, but the community they've collected to join them in supporting those efforts, ranging from the Grub Stake Fund to the Gates internships to the Gates Institute you're going to hear tonight. And also been motivators, because we like to think we're moving pretty fast. Diane thinks we're moving pretty slow and reminds us of that frequently. And it was her vision and the commitment of her family foundation, her and her brother John, and a matching commitment from the chancellor to put together funds of $200 million to rapidly accelerate this effort over the next few years. So we're really looking forward to what the impact of this program is going to be. We're fortunate that Terry Fry has been named as the first center of the Gates Institute, succeeding Dr. Roop in the leadership role, who will remain on with an important leadership role in the program. He can't be with us tonight because he's recovering from a fairly major surgery, but remains an important force behind this. I want to thank Diane Gates-Wallach, the Gates family, and all the members of the Gates Advisory Board and the Gates Scientific Board for all their support through this process, and ask that you recognize them now with a round of applause. So the whole focus of the Gates Institute is to take advantage of patients' own cells and re-engineer them and modify them in ways that either produce a therapeutic benefit, as you're going to hear about tonight, or helps patients regenerate tissues. With this establishment of this institute, we hope to accelerate the progress on all of those fronts. We have a number of open clinical trials on this campus now, primarily focused on patients with hematologic cancers, but many more in the pipeline. As somebody who's in the tail end of their medical career, I'm a little jealous of our medical students who are at the beginning of their medical careers, because the next decade in medicine is going to be tremendously exciting. These therapies are really a game changer. So one of the pleasures of being the dean here is we have great faculty, and you're going to get to meet a few of them tonight. So I want to start by introducing Dr. Terry Fry, who's the newly appointed director of the Gates Institute. We were fortunate to recruit Terry from the National Cancer Institute almost 5 years ago now, 2018. You'll hear about some of his work tonight, and as well as other faculty members whose pictures you see up here, Mike Veneris, Amy Keating, Manali Kamdar, all physicians on our faculty. And I especially want to thank Larry Fierstein, a patient who has offered to share his experiences with you tonight to get you to realize firsthand what the impact of these therapies has been. So Dr. Fry, in addition to being the director of the Institute, is the Charles C. Gate Endowed Chair and also the director of our cancer immunotherapy program and our cancer center here on the campus. And I will now depart the stage and turn it over to Dr. Fry. Thank you.
So thank you, John, for the really perfect introduction and kind comments. And, what I'm about to say is something I really mean. It is an honor to be standing here tonight as the incoming director of the Gates Institute. Honestly, when you think about, opportunities in your career, it's rare to have one that you look at as, really a dream. I can truly say that standing here tonight is a dream of mine to be leader of this institute and to really realize the vision of Charles Gates, which is something that I certainly share. So I thank Diane and the Gates family for the opportunity. And what we're going to talk about tonight is, as John mentioned, a therapy called CAR T-cell therapy. And my job here in the first 10 or 15 minutes is just to orient you as to what this is. Many of you have probably heard about it. If you search the internet about this therapy, there are things like talking about using HIV to treat cancer, et cetera. But what we're going to try to do is to really give you insights into what it really is. And as John said, I mean, it's as the leadoff speaker, I will tell you that I am definitely not the highlight. The highlight is what you're going to hear later in the presentation. But just to kind of frame this, I'm going to start sort of to broaden this and think about where immunotherapy as a broader therapeutic fits into traditional cancer therapies. And as many of you probably know, unfortunately, probably too many of you, the standard treatment paradigm for cancer is local treatments, surgery to remove tumors, radiation to radiate local tumors, and in situations where the cancer is spread, administration of what really, quite frankly, are poisons, cytotoxic chemotherapy, to try to destroy the cancer cells. And certainly we recognize that that particular approach is not very selective, as there's a lot of toxicities that come from that. And so what we're talking about tonight is really the ability to harness the immune system, to treat cancer in a way that is, hopefully more selective and has the ability to control tumors that are distributed throughout the body. And it turns out that the T cell, the cell we're going to talk about tonight is something called a T cell. It's one of our white blood cells. Think of it as a member of an army that essentially circulates throughout the body, trying to identify either damaged or cells that are infected with something like COVID. And when they find those cells, what they do is they destroy them, and they do that by one of the ways they do that is by secreting things that actually literally punch holes in the damaged cell that results in essentially an explosion of that cell. And so in that regard, these cells are very powerful at destroying cells that they're able to identify. And it turns out that for certain types of cancer, T cells actually have the ability to see those cancer cells. In skin cancer, lung cancer, there has been remarkable success by administering drugs called immune checkpoint inhibitors that essentially just take a little bit of the brake off the immune system and essentially unleashes the potential of these T cells to destroy the cancer cell. Unfortunately, it turns out that for certain types of cancers, the ones we're going to talk about tonight, T cells don't really see those cells very well. And so what we're going to be talking about is a strategy to essentially take this T cell that is not able to recognize those cancerous cells and essentially introduce a gene called a CAR gene that results in a new protein on the surface called a CAR that allows that T cell to now recognize these cancer cells. And so in many ways, that protein is not physiologic. It is a bit of a, for those of you that are old enough, it's a Buck Rogers approach. And I would say that if the people that are sitting around in the room thinking about this, it was sort of a crazy idea. But it works, and you're going to hear about that tonight. So this therapy is very different. It's a living therapy, and I'm not going to walk you through this because one of the subsequent speakers is going to talk you through, Mike's going to talk you through the manufacturing. But this is a cell that is taken outside the patient, it's modified, it's frozen, and it goes back into the patient, and it's alive at that point. And we know that in some patients, these cells can persist for 10 years inside the patient, providing in certain cancers, a needed active surveillance for some period of time. So the first description of this was in this paper. This is actually what I think is the first description. There's another paper that was published a couple of years later by Zeligesher. And there are hand drawings in this paper that describe the gene itself and the crazy construct. And this was in 1987 when this came out. And it took about 15 years or so, and a number of physicians took this into patients, and it didn't work. And this is actually not an uncommon theme in the context of development of a novel therapy, is that the first time you try it, it doesn't work as well as you think it might. There's a lot of surprises when you go into patients. And so Scientists, researchers took this concept back into the lab. They tinkered with it a little bit. And a number of years later, 2010, the first publication of what we now recognize as a CAR T-cell therapy. And so this is, what you see in the bottom there is a slide of a lymph node. And that is actually, the brown in that are all tumor cells. And that patient received a single infusion at I was actually at the National Cancer Institute when this patient was treated. That patient received a single infusion of these CAR T cells that you're going to hear about. And a couple months later, all the brown is gone. And this was the first report of the ability to use this therapy to really induce dramatic responses in these patients. This example that I just showed at the NIH was happening at a time when there were multiple trials like this that were being conducted throughout the United States, all at academic institutions in the National Cancer Institute. All with slightly different variants of this, but they were all seeing the same thing, which was that in leukemia, pediatric leukemia, about 80% of children who were refractory to all other therapies, many of whom had received bone marrow transplant, were achieving complete remission. That is, no more cancer cells detectable. And in the lymphoma setting, it was about 50% of those patients ultimately achieved what's called a complete response at about 90 days. And this led to ultimately an FDA approval in 2017, really the first approved gene therapy. And as a pediatric oncologist, I say this every time I give a talk. This drug was first approved for pediatric cancer. That's almost unheard of, that the first approval is actually in pediatrics. And these are the headlines of a couple of papers at the time that some of you might recognize. This led then to really a whole series of approvals in adult lymphoma with a couple of different CAR drugs, and then ultimately more recently approvals in a different type of blood cancer called multiple myeloma. Along with that, there has certainly been a tremendous amount of enthusiasm and investment external to academic institutions. This is just a slide just demonstrating sort of the activity in terms of investment in this area from the Alliance for Regenerative Medicine. And so, almost over 2,000 ongoing clinical trials as of June of last year. Investments first half of last year totaling more than $6 billion. So obviously an area of very, very active investment and enthusiasm. And what I would say is that if you look at sort of this field, in many ways, the CAR T-cell activities have really led to, at these academic institutions, that's really what led to the development of many of these biotech hubs. This is just a list of the institutions that are actively running CAR T-cell trials, as John mentioned, including our own at both of our hospitals on this campus. And if you look at a lot of these cities that are familiar to you, you will see that in this report, not ours, this is from the Alliance for Regenerative Medicine, this is what they have identified as being the landscape of the cell and gene therapy hubs. And what I would like to see is that the next dot on there is actually located where we are. This is my last slide. So the Gates Institute. So John mentioned the Gates Institute, and really just a word about what we're hoping to build here. I think that one of the phrases that Charles Gates used to use, and Diana's mentioned this many times, is that he liked to make sure that you could remove the friction from progress. And I think in many ways what we're trying to achieve here is taking what is a complicated scenario where you've got to take a scientific idea, you can get it to early discovery, but getting it, and Mike's going to talk about this a little bit, that leap from that stage into clinical testing in humans is a big one. And it requires a lot of capabilities that most academic institutions, probably don't build out because it's hard to fund with standard grant funding, et cetera. And so hopefully what we're going to be able to build out here at the Gates Institute is an entity that's really going to facilitate that translation of great science that's happening on this campus into patients. And so with that, I'm going to introduce a colleague and really a friend of mine, Mike Veneris. It's a pleasure to introduce Mike. I asked Mike what I could say about him that was embarrassing. I'll choose not to, but Mike is a bone marrow transplant physician, a professor of pediatrics, at the Children's Hospital in the Barton Family Endowed Chair of Bone Marrow Transplantation and Cellular Therapy. And this is how I'm going to embarrass Mike, is I'm going to introduce him by start showing you guys a little bit of a video that I think gives you a flavor for what we're doing.
One of the unique aspects of CU Anschutz is that we have a world-class children's hospital and a world-class adult facility coupled with laboratory research facilities and the biomanufacturing facilities, which brings together a group of very different individuals, clinicians, doctors, researchers, and the good manufacturing facility, all to create therapies here on campus for patients. To make CAR T cells, what we do is we collect blood cells from the patient. They're then shipped to the biomanufacturing facility where they are genetically modified using a weakened HIV-based virus that brings in a gene that then allows that patient's T cells to recognize and kill their leukemia. This type of research recently has really been transformative for patients with both leukemia and lymphoma, and we're testing a slightly different version of the CAR, which is the chimeric antigen receptor, to see how it works in our patients. I think the most powerful thing is it really is changing the practice of medicine. Instead of giving multiple rounds of chemotherapy, this is an entirely different approach to how patients will be treated with cancer. And the optimism, both at a university level and at a national level and at an international level, is just incredible. I mean, I've not seen this type of optimism in over the 25 years of my career.
So I'll thank Terry for showing that video. It's really one of the most difficult things to see yourself on the screen like that. And I think I'm wearing the same shirt, by the way. Yeah, so as Terry points out, my role here for tonight is to really explain how to go from promising research in the laboratory to a patient. And so I thought I'd start with the experiment that we did in our own lab. I think on the left-hand side is this green ball, which is the tumor cells, and the red dots that you see are cells that are actively dying. And if you look, we tested a CAR that was directed against a really promising antigen that's really only on cancer cells. And this is really the standard approach. And then we modified this in our lab, right? And so we changed these cells to now not only home to the tumor better, but also kill the tumor better. And so we're really excited about this, and we want to get it to patients. So how do you do that? And so, again, that kind of starts with the lab and ends with patients. But there's this whole area that we've listed here as scientific roadblocks, right? And this is really called the valley of death in biomedical research. And really the Gates Institute kind of, our vision is to really bridge that valley of death with these promising observations and to really take a car and drive it to the patient. And that was not my idea, by the way. But all kidding aside, this is a major problem. There's incredible research going on at this institution, not only here at Anschutz, but at Boulder and in Colorado Springs and in Denver. And how we translate this work to patients is really hard, and that's what the Gates Institute is going to allow us to do. And so what are some of these roadblocks? Well, I think the first roadblock listed up here is the discovery and the preclinical observations. I'll point out that the National Cancer Institute funds our research, and only about 9% of all the grants get funded. That means that 91% of all these great ideas never get to patients, or never even get to the preclinical phase. Like I said earlier, I think that there's a lot of clinical promise on this campus that's not really realized, because the people in the lab are kind of doing research sometimes for discovery, but they don't really understand how to apply that research to patients. And I think we really feel like we have a role to kind of bridge those laboratory observations to bring them to patients. And if you publish those laboratory observations before you patent them, then you lose a huge opportunity to actually ultimately sell that to pharma in the end, as part of our sustainability program that Diane has pushed us so hard to think about. As far as the manufacturing goes, again, we have a good manufacturing practice facility, the Gates Biomanufacturing Facility, and we're lucky enough to have this facility. But this, like you saw some of the images on the screen, but think about like Silicon Valley and wafers and stuff like that. They're clean rooms, they have to be extremely clean. And so you have to be able to take the stuff that you're doing in the lab, where you're doing it with, like, let's say 500,000 cells, and get it all the way to 2 billion. Just to put that into perspective, that's like Denver or Colorado, perhaps, all the way to China population, right? So you really need to be able to scale these things up. You need to be able to, all the stuff that we use in the lab is not necessarily clean enough to use in patients. So you need to know what you can and can't do in this GMP facility, and you need to partner with those people. As far as the regulatory work goes, patient safety is a huge issue, right? And compliance with patient safety is a huge issue. So this field is complex. It's moving quick. So the cell and gene therapy landscape and the rules around this are really challenging. And I think back, like when Terry and I were like interns, there were these gods who saw a patient, was interesting, weird disease. They didn't know what it was. They drew blood. They took it back to the lab. They cloned the gene. They figured it out. They then created a therapy, and they gave it to the patient all themselves. Those days are long gone. There is no way that there's a one-man show. This is the teams that get these cells into patients are probably in the range of more than 100 people. I mean, it's really that much work to go from an idea all the way to a patient. I think the other thing is that patient safety and how you monitor the safety and how you monitor the activity of these cells is something that we're really challenged by. How do you design a clinical trial to prove that these things are not only safe but perhaps active? And so these are some of the roadblocks that really we're hoping to remove with the Gates Institute. So from the discovery and preclinical arena, again, the Gates Institute is providing funding for promising projects to really, again, bridge that gap and get these projects from the lab bench into the patient. And again, we're trying to go out and really find projects that are of that are really kind of are novel and can change the practice of medicine, which is our goal. This middle thing here, taking the next step, it turns out that some of these projects have been in people's labs for 10 or 15 years, and letting go of a project and giving it to someone else is actually not so easy. It's like letting your kid go to college. And so again, the way we plan on doing this is enticing them with the people over here, project managers and people with expertise, because if you don't have this, you can't get this into patients, simply put. And then lastly, we try and educate these people in the process of how you go from your laboratory experiment all the way to a clinical trial. CAR T manufacturing is a really complicated process, and in some ways it's not unlike the ice cream maker and Chopped. And what I mean by that is that I think many in the room have probably watched Chopped, and they give you some ingredients, and you have to make a dessert at one point in the show, and you have to come together, and you get all your stuff, and you put it in the machine.
But occasionally, you don't turn the machine on or you don't turn the refrigeration on, and you run into problems. And I think that's really what it's like to make CAR T cells, right? And what I mean by that is it really requires a group of skilled laboratory people. They're not the laboratory people in RC1 or RC2, the research, basic research. These are people who run the GMP facility. There's probably over 50 people who are dedicated to this process and really make it work and make it perfect. And again, in some cases it works, if you don't do it right, you can end up with the chopped analogy. But really, as Terry alluded to earlier, the way you do this is that you start with the patient and you collect blood cells. So you put in 2 large bore IVs, usually in their arms, but sometimes in other areas, and you collect the cells through a process which is called apheresis, where blood is being drawn, some of the white blood cells get siphoned off and then get pushed back into patients. Once that product is isolated, it's then sent to the GMP facility where the T cells are purified, and then the cells are infected with a virus, an HIV-based virus that actually has the CAR gene in it. And then the cells are grown, they're quality control tested, and they're ultimately infused into the patient. So as far as the next kind of roadblock is those regulatory roadblocks. And this is really what I think of when we start with our campus. There's this crazy line, and then there's the FDA. So everything we do has to be approved by the FDA, and the interactions between us and the FDA are really complicated, and they're nuanced. And I will say that if we don't do this right, we could lose all the federal funds that we have at this institution. So there's a lot of pressure around this. And obviously, we want to do it right for many reasons. But doing this is resource intensive. You need people who understand how to interact with the FDA. These interactions are legally binding, and really it's the only way to do this. So again, with the Gates Institute, we are like building teams to actually interact with the FDA. But I'll give you an example. Like if we want to do a research study today, and we submit it today to the FDA. We've got the binders. There used to be one of our colleagues, his name is Malcolm Brenner. He's not the tallest guy in the world, but he used to show like a picture of him with about a stack of papers about this high, which is what it took to get a project submitted to the FDA. So it's a huge amount of work. But anyway, if you submit it to the FDA today, 28 days later, if we don't hear from the FDA, we're good to go. So they don't say you're good to go necessarily, but they just don't say you're not good to go. So it's this kind of anti-speak that you have to understand with the FDA, and it's just the rules. You have to understand how to use them. And then I think lastly, obviously, is patient safety, right? And how do you, this is a totally new class of drug. As you heard Terry say, these are living drugs. So if you think about it for a minute, if you look at penicillin, everybody's taken penicillin in this room probably. In the red arrow, you give a dose of penicillin, the blood levels go up, and then they go back down. 6 hours later, give another dose, it goes up, down, and then when you stop taking it, it goes away. CAR T cells, on the other hand, when you give the CAR T cells, you infuse them, and then over the course of 2 or 3 days, what happens is that they expand dramatically, up to 10,000-fold in the patient. And when they do this, patients can really get sick. And the clinical trial that Terry talked about, where it was first approved in children, we participated in, and in half of those patients actually went to the intensive care unit. Thankfully, no one died from it, but they became incredibly ill when we gave these CAR T cells in the early days, because we didn't understand the side effects, number one, and we didn't understand that a high disease burden puts you at risk for really getting sick. But those T cells expand, they clear the leukemia or the lymphoma, and then they come down to a slightly lower baseline, not down to 0, but they're there, and we can detect them in patients. And then all of a sudden there's a blip. And we wonder, is that because maybe it missed a few of those leukemia cells, and all of a sudden it found some, and it killed them, and expanded, and then it shrunk back down? And there are these little blips that happen from time to time. But understanding the biology of this is really quite amazing and quite interesting. And as Terry alluded to, some of the very first patients who have been potentially cured from this, you can still find these cells, these genetically engineered cells, 15 years later in these patients, which is really quite stunning. I mean, it's really quite amazing. So obviously, one of the other things that we really
would be remiss is to not talk about our interactions with our two clinical partners. UC Health and Children's have been fantastic partners in this. They allow us to recruit patients to these studies. These are first-in-human studies. I mean, these are really high-risk studies, and I think that the partnership has been phenomenal and that we could not do it without that, and we're really lucky because we're all centralized on this campus. And then lastly is this picture. And this picture, I think, really speaks to the success of the study. And you can see here, our first study was in 2020, our second study in 2021, our third study in 2022, and we have one that's due to open Q2 of this year, so this upcoming year. And you know, we talked about this a lot, and the excitement of doing these types of studies as a physician is really, really quite amazing, right? Because when you watch that thing kind of go down the IV tube and you know this is the first patient to ever get this, this is really like exciting for us as clinicians, but these patients depicted here have capes, and I think the capes are really because they're incredibly brave, because sometimes they're the first person receiving this, or really early. And so I think that really kind of acknowledging the patients is a huge, it's a heavy burden that we bear, like that we do no harm. That's our first rule. I think lastly, I'll just point out that the top 3 trials were for patients who were chemotherapy refractory, leukemia, or lymphoma. And really what that means is that if you have chemotherapy refractory lymphoma, you have a lifespan that's measurable in weeks or months. So all of the patients who were treated on this really were in dire straits. And without going into the details, the majority of these patients are still alive. I mean, it's really stunning and really moving, and it's really an honor to be part of that. I think the last part, this bottom part, is a little different. So this bottom part is a study looking at CAR T cells in adults with leukemia. And without going into the details, after the first month or so, we know whether that leukemia has been cleared or not. And if the leukemia is still there at 2 months, we know the chances of that patient relapsing are really high. Still, we do the thing that we do, and we give chemotherapy, and we hope for the best. And instead, what this study is going to do is really take those patients, pull them out of regular care, and offer them CAR T cells with the goal of not suffering through literally years of therapy only to ultimately relapse. And so, like I said many times previously, I think by the end of my career, I hope I will have a medical student where there's a brand new kid with leukemia, and I kind of walk up to that student and I say, I remember when we used to give chemotherapy to these patients. And that's really, I think that's really our goal, to change the practice of medicine. So with that, I will introduce my dear colleague and friend. She is the medical director of the Pediatric Bone Marrow Transplant Program at Colorado Children's, and she's gonna share with you some of the patient experiences on one of those pediatric studies.
Thank you so much, everybody, for coming and enjoying our talks about transforming healthcare. As Mike just mentioned, it's mind-bending how much change we've made in such a short time. And I'm hopeful to share with you some of the experiences that have happened on the pediatric side. And I thank Drs. Fry and Verniers for giving us all this opportunity and for setting the stage of what CAR T therapy is. So my goal with tonight's talk is to kind of go through 3 main areas. People often think of cancer as an adult disease and a disease that affects only adults, but in fact it has a large impact in our pediatric population, and particularly leukemia, and we'll go over some of that. And we're going to go over the role of immunotherapy to treat leukemia, especially for those patients who aren't responding to our standard therapies. And finally, we're going to run through some of the current barriers that we still confront with immunotherapy and how the UCD19 CAR T program here on campus really overcomes some of these barriers and leads to lives saved. So cancer is the #1 cause of death in children related to disease. I don't think people comprehend that, but it is the reality that we deal with every day in pediatrics, and it's why myself and Terry and Mike do what we do. Leukemia and lymphoma, along with brain tumors, make up nearly 2/3 of pediatric diagnoses, and leukemia and lymphoma together are well over a quarter or a third of all pediatric cancer diagnoses. So a very large impact for our patients. And a child that dies of cancer on average loses 68 years of life, which is a tremendous amount of life lost, and everybody would agree is unacceptable. And so one of the things we've really tried to look at is how can we make this better? And we have made really great strides in survival. So back in 1975, just a few years after I was born, the survival rate for acute lymphoblastic leukemia was only about a third. So a third of patients who got diagnosed with ALL made it, and 2/3 died of disease, and typically within the first few months. And over many years with many trials and adding together different types of chemotherapy along with radiation and bone marrow transplant, we've been able to bring that number up to 87% in the last SEER analysis, which is an amazing amount of progress in just a few decades. However, there's still a good bit of patients that are lost to disease. So what happens for these other 10%? So as Mike alluded to, prior to the advent of immunotherapy, survival for the other 10% was dismal. We really had no options. We used cycles and cycles of poisons, things like chemotherapy, radiation, and even bone marrow transplant. But the intent at that point wasn't to cure the patient, it was to prolong their life and give them as many months as possible with their family and loved ones. But we weren't intending to cure, because if you're not responsive to our therapies, you will eventually die of disease. So cell therapy came along and offered a new way to effectively target leukemia cells, which has really been groundbreaking and changed a lot of patients' lives. So as alluded to, tisagenlecleucel, or otherwise known as Kymriah when it became commercially available, was the first CAR T introduced for leukemia. And initially in the trials that were developed by CHOP and then Novartis, it was trialed in actually pediatric patients, which is a little bit of a different approach. And the patients had already failed either transplant or up to 8 previous cycles of different types of chemotherapy. So these are people that they'd tried just about everything and they were completely unresponsive to everything. And when they gave them tisagen or tisacel, 93% went into a remission, which is unheard of because previously it was 0%. And so everybody, we were very surprised and we were very excited by this result. And we opened up CAR T centers throughout the US and throughout Europe to deliver this therapy to children. However, there remain ongoing significant hurdles that I'd like to discuss tonight that with the UCD19 program we've tried to overcome. And those are timing, toxicity, and the worst of all, I think, is relapse. So the timing of CAR T cell therapies and really the goal of speeding up the process. So we went through some different ways that the cells get made, and it seems all quite easy, right? You take the cells out of the patient, you put the gene into the patient, you give the cells back, and voila, they're cured of their disease. Seems simple, seems amazing, can take a very long time. So in the initial days when this therapy was introduced, insurance approval was taking upwards of 3 months, and some patients never were able to receive approval at all. These are pediatric patients with relapsed and refractory leukemia. Then you get insurance approval, you have to take those cells off, you have to send them to the other side of New York, and 6 weeks later those cells return. And in fact, in early previous trials, more than 10% of children died before they ever received the cells back, and this was without needing insurance approval. So you can imagine what the barriers are in the setting of commercial and waiting for insurance approval. With the CAR T UCD19 program here, we've been able to take this minimum of 6 weeks and transition it to 17 days, and it's actually improved with each patient that we've put on trial. And the first one was a bit of an outlier, and now it's more like 13 days. So 2 weeks to go from cells coming out of patient to patient starting therapy in anticipation for CAR T. It seems maybe not a big difference, 6 weeks, 2 weeks, 3 months, 2 weeks, but it's a tremendous difference. So these patients have active leukemia. They are actively dying of leukemia that is untreatable. And so to come up with some magical therapy that would potentially treat them for 3 months can be impossible, hence why 10% of patients are dying. So this is a tremendous change and something that I think should not be underestimated. Another thing that we alluded to previously is the toxicities of CAR T. So with every good, there is bad, and with CAR T, there's a lot of bad that can be very dangerous and something we've learned to manage and monitor for. The two biggest ones are probably cytokine release syndrome, which we call CRS, and the other is neurotoxicity. So CRS is you take all these cells that are all activated and ready to go and ready to fight, and you throw them in the patient who has a large disease burden, and those cells go crazy, and they get activated, and they expand, and they cause a major, massive immune response, which is great. That's what we expected, that's what we wanted, but it can be dangerous and life-threatening. And in about 15% of patients currently, we're still seeing severe grades of CRS. When I say severe, high fevers is greater than 105 degrees Fahrenheit, so that, just to put that in perspective, these are very high fevers. Low blood pressures, so much so that you end up in the ICU on medications to maintain your blood pressure and maintain blood flow to your organs. And low oxygen in your blood, which often requires putting a tube down to help people breathe. Neurotoxicity, on the other hand, is not as well understood. It makes sense that when the immune system gets activated, you get lots of inflammation, and we've become really good at managing that. But neurotoxicity is a little less well understood and ranges quite a bit. It affects less of our patients. Some get a simple headache, and we go, oh, that's too bad, take some Tylenol. And some get seizures and end up in a coma and are unable to speak. It's typically self-limited. It goes away on its own, which is a little bit scary for families and patients. But in some of the trials in adult lymphoma actually did lead to enough brain swelling where patients died from it. Luckily, we've not experienced that in our leukemia trials, but it's two things that we watch very closely for. So you can see our rates are somewhere in the mid-teens to 10% for these severe neurotoxicities, but with the UCD19 experience, we've had no CRS of significant clinical degree or no neurotoxicity either. Of course, we're still testing higher and higher doses, so it's likely as we go to higher doses that we might see more of this, but it is one of the things that we've been able to monitor very closely, and thus far we're pretty impressed by our early safety signals. And then the final horrible, terrible T, relapse after Tisacel. This is seen with most CAR T trials. In the pediatric experience, it's about 50-50. So 93% go into an immediate remission. We're very excited. We're very blessed by that. But 50% of those patients will go on to relapse their disease. 50% of those patients relapse their disease because they lose their CAR T. So that makes sense to us, right? The CAR T was working, it stopped working, it was rejected, and the patient relapses. Another quarter of those patients will actually, their leukemia will evolve. So the leukemia will start to grow and stop putting the protein that the CAR T is responding to, the CD19 protein, stop putting it on the surface. So no longer is it signaling to that CAR T, hey, come and attach to me and attack me and kill me. It's now undercover and can go on and rapidly relapse unchecked. So our experience so far with relapses has been, and with remission and relapse has been really great. So on the pediatric side, we've had 6 patients go on to trial. And all 6 patients went into an immediate remission. And again, as a reminder, all of these patients were refractory to initial upfront therapy of multiple cycles. Just to speak a little bit more detail on each of these lovely patients. Patient number 1, to me is the biggest hero because he came in not knowing what he was getting, first in human. We infused him with these cells. He had already been admitted to an adult metro area hospital for about 9 months straight, had received multiple rounds of chemotherapy, had no response, or sometimes would go into a half a remission for a week or 2. And he did great. He went into remission at 1 month and was MRD negative. Unfortunately, he succumbed to his disease a year later when his leukemia stopped putting that protein on the surface. And the CAR T, while present at the time of his leukemic relapse, no longer recognized his leukemia, and he died of his disease. Patient number 2 was a lovely patient who was ineligible for either commercial CAR T or bone marrow transplant, and had multiple relapses of her leukemia. They kept trying to push it off and do those things and make her life last as long as they could. And we were able to offer her our trial CAR T product, and she went into remission and is 18 months from that infusion and still doing great. Patient number 3 was a patient who came to us and had a massive infection in his leg and was unable to tolerate normal chemotherapy or radiation. And so his options were treat the infection and not treat the leukemia, or nothing. And so he came to us and said, well, can I try this? And we said, we sure can try. And he is 12 months post that infusion and doing great. He was hoping to join Larry tonight to discuss his experience with you all, but this week he decided to get married. So he is off getting married. He graduated high school and then got married, all in a few weeks' time. Patients number 4 and 5 are both post bone marrow transplant. So they had already multiply relapsed. We got them into remission. We took them off to very intensive therapy, and both of them relapsed after transplant. And we infused both of them with CAR T, and they are doing great. One is at 1 year post that CAR T infusion, and one's at about 6 months, and both doing well. And our final patient was just more recently infused. She had also had completely refractory disease, did not respond to anything, responded to CAR T, and then went on to bone marrow transplant for a consolidative attempt at remission, and is doing great post-transplant, and was just seen in clinic this week. And we have patient number 7, hopefully coming up for infusion by the end of the month, that I'll be able to update y'all without too much time. So this is a Herculean effort to put a trial like this together, and takes years and years of people and teams and groups supporting you and cheerleading you. This does not nearly represent all the people that participated in the writing or the activation or the continued use of this trial, but hopefully represents the different teams that have been so important, both campus-wide and nationwide, to get this going. And with that, I'm going to introduce my colleague across the street and adult compatriot, Manali Kamdar. She's the head of the lymphoma program for the Hematology and Bone Marrow Transplant Division at CU Medicine, and is going to talk to you about cell therapy for lymphoma in adults.
So very good evening to everyone. Thank you so much for being here to hear about our experience with our homegrown trials, as I call them. So just to walk you a little bit through what lymphoma is all about. Lymphoma is a blood cancer of one of the fighter cells of your body called lymph cells. And lymph cells basically go rogue. We don't know why that happens. And because it's a blood problem, it can go anywhere. We don't really put that much emphasis on stage when we talk about lymphoma, because 85% of patients are in stage 3 and 4. But usually the prognosis of lymphoma is more around the subtype of lymphoma than the stage of lymphoma. So alluding to the subtypes then, lymphoma is not one disease. It's actually made up of nearly 100 different subtypes, 107 to be precise. As a result, it's really important to understand which subtype am I dealing with in order to deliver the best care for these patients. Just to walk you through a basic algorithm, which I usually tell my patients when I see them in clinic with lymphoma. They're divided into either non-Hodgkin lymphoma or Hodgkin lymphoma. Non-Hodgkin lymphoma afflicts 80,000 patients in the United States every year. Non-Hodgkin is further divided into either B-cell non-Hodgkin or T-cell non-Hodgkin. B-cell non-Hodgkin makes up for nearly 85% of those 80,000 patients, and that B-cell non-Hodgkin lymphoma is further divided into 3 buckets. I won't belabor the time here, but to clearly just allude what we are really getting to is the first bucket, which is aggressive B-cell non-Hodgkin lymphoma. Usually, chemotherapy is the mainstay of treatment. About 50% of patients can be cured with upfront chemo. However, 50% will not. And these are the patients who used to previously succumb to just chemotherapy after chemotherapy, until cellular therapy made a huge difference in the survival of these patients. The second bucket is a bucket called the indolent lymphoma, or a slow-growing lymphoma, which basically means that patients are diagnosed with this lymphoma or disorder, they live with this and pass with this. So treatments that we really deliver are around making sure that the quality of life is preserved. We want them to have everything they want to do in life without really coming in the way of their normal day-to-day aspects of activities of daily living. As a result, therapies here are really geared towards making sure that these patients are in remission for a very long time. But the unfortunate truth is that these patients are very susceptible to relapse after relapse. As a result, we do need to develop more treatments that can give them a long remission. And the third bucket, which my dear friend, my patient, and almost my family, Larry Firestein, is going to talk about, is mantle cell lymphoma. Mantle cell lymphoma is extremely unique because it actually harbors both. It has the biology of a non-aggressive lymphoma, but it can swap and become an aggressive lymphoma. As a result, these patients are not only presenting with a lot of symptoms, but they also are very susceptible to multiple relapses over decades. And therefore, it's extremely important that we develop treatments for this lymphoma. Immunotherapy has evolved radically over the last 5 years. But it took many, many decades to get to where we are. And it cannot happen without clinical trials. Transplantation is the first immunotherapy. Vaccination is the second immunotherapy. And over time, I will say CAR-T, to actually get to where it is, it took nearly 3 decades. And at this point, we definitely have a plethora of options, which is all great. But it's important to understand sequencing to figure out which is the best treatment for the patient, which will put that patient in a remission for the rest of his or her life. This is a patient of mine, Cellular, lives in Colorado Springs, came to me and said, only if you have something miraculous am I going to try it. And I'm not going to try chemo. He had tried 7 lines of chemotherapy. His abdomen was distended, as you can see on the left side here. It was a 25-centimeter mass in the abdomen. And I said, I don't know about a miracle, but I think I do believe in God and science. And if I was to combine the two, I think I have a clinical trial for you. And that is our first experience with CAR T-cell therapy on campus. We proceeded forward with the clinical study. 90 days later, as you can see on the right, the entire tumor melted away, and he was in a complete remission. He remains in a complete remission, and he has gone back to living life. He's actually decided not to come back to me, and I'm totally fine with that. So since the clinical trials got instituted, there have been 3 CAR T-cell therapy constructs that have been FDA approved. And with this, 40% of patients who would have previously succumbed to their lymphoma are now able to live a normal life. So that's pretty good. But of course, we need to do more. We need to develop more effective treatments. Our plan is to cure 100% of patients. So previously, the algorithm used to be that if anybody came to us who had relapsed after initial chemotherapy, we used to assess them for transplant fitness. Bone marrow transplant can either be from self, meaning auto, you give your own cells back to you, or a donor. Auto transplant had a potential to cure fit patients about 40% of times. And typically, if auto didn't work, we didn't have any options, but then CAR-T came along, and those 40% of patients who failed an auto got cured by CAR-T. Now, clearly, not all relapses are created equal. So auto typically works very, very poorly in patients who relapse within the first year of having received chemotherapy. So I'm going to introduce to a concept in medicine called the Kaplan-Meier curve on the right. Any curve that looks like that means the survival is dismal. And for patients who relapse within a year of getting chemotherapy, the survival was less than a year. To be exact, 6.3 months. And therefore, it was intuitive that if CAR-T works in patients who fail an auto transplant, could we actually bring it forward, compare it head-to-head with an auto, and figure out if we can actually make auto an extinct entity? And this is where 3 large global randomized studies were launched which compared autotransplant to CAR T-cell therapy across the globe. And I will have to say, the campus was a leader in the study called the Transform study. We enrolled a lot of patients, not only from Colorado, but also from neighboring states of New Mexico, Arizona, Wyoming. And Transform study was a positive study. I just taught you about Kaplan-Meier curves, and you can see that the blue curve is over the black curve, which means CAR T won hands down over autotransplant. And on June 22nd, 2022, autotransplant became extinct for patients who relapsed after initial chemotherapy within the first year of receiving treatment. For these patients now, we do CAR T-cell therapy. And this came out of what we did at the University of Colorado. In terms of toxicities, though, we cannot turn a blind eye to that. Remember, CAR T-cell therapy is basically engineering a living cell. You put them in the patient's blood again, and they unleash upon the tumor. As the tumor dies, they release certain substances which are called cytokines. It can create what we call an inflammatory storm. It can lead to low blood pressure, fevers. It can lead to neurological toxicities. In addition to that, to manufacture CAR Ts with the existing FDA-approved products takes 4 to 5 weeks. Now you may imagine cancer is not waiting on us. And as a result, we need products that can be manufactured rapidly, that do not carry these toxicities, that are efficacious, probably much more efficacious than the existing CAR T products that are FDA approved. The one side effect of CAR T is diffuse inflammation. Your fighter cells that have been manufactured in the lab, when they get infused into the patients, they just become so active that they inflame every single organ. And that can cause a lot of toxicity. This is another patient of mine on a clinical study. He has a 35-centimeter tumor in his right thigh. You can see that on the left. Went on to the clinical trial. Day 90, the tumor vanishes, but his spleen becomes enlarged, and his cells, in terms of his hemoglobin, his red cells, platelets, they are all low, and he becomes transfusion dependent. This is because of the massive inflammation that happened. I remember that night when I frantically called Terry Fry, and I was like, what do I do? And he gave me advice. He told me about immune suppression agents, which I had to use. So of course it's a team effort. One day I was just counting the number of members that are needed to take one patient through CAR T. And to be precise, if I just look at the clinical personnel, I'm not even talking about the lab. On my side of the game, it's 107 people. So clearly, it's a huge undertaking. But for a patient who has failed 10 lines of chemotherapy, to get a response as mind-boggling as this is worth all the effort. We're now in a unique space where I told you that 40% of patients definitely have a great survival with CAR T. What happens to the remaining 60%? With the current FDA-approved molecules, patients definitely go into a remission, but about 60% will relapse. And you can see on the right, based on that curve, the overall survival for patients who relapse after CAR T-cell therapy is very dismal. As a result, the new unmet need in the field is really developing either effective CAR T-cell therapy products that are more effective than what we have, or developing treatments where patients can be treated who have received prior CAR T-cell therapies. And this is where the genius of Terry Fry, Matt Seefeld come into play, where we are really looking at mechanisms of figuring out how to get smarter than the cancer. You all know that the existing CAR T products target one protein on the lymphoma cell called CD19. Now the cancer is smart. Definitely initially it might just get hushed, but it might develop a resistance where it might lose the ability for the CAR T to recognize that CD19 on its surface. So then, can we trick the cancer cell and make a CAR that can target two proteins on the lymphoma cell? This is the idea behind the 1922 CAR T bicistronic trial that we have on campus that is led by Dr. Fry, Dr. Veneris, and us on the adult side. It's also exceedingly important to decrease the manufacturing times. And as Dr. Keating alluded to, the UCD19 CAR T trial manufactures product less than 17 days. So to give you a little glimpse into how have our patients done on our homegrown CAR T UCD19 trial, these are my 7 patients. I will tell you that the median lines of treatment have been at least 5. They have failed 5 lines of chemotherapy at least. The median age was 61, but it ranged from 50 to 80. Median follow-up, meaning for how long they are now currently here, is about a year. And at this point, 6 out of 7 patients have achieved a complete response. Interestingly, we did not see high-grade cytokine release syndrome or neurological toxicity. And manufacturing was successful in all patients. This is clearly a wonder, in my opinion. In terms of toxicity, I think the biggest toxicity that our patients had was boredom. They were so bored at the hospital that every day they were itching to just get home. So at this point in time, I think we have made really astounding success thanks to the support that we have received from leadership to be able to run clinical trials here. It is a huge undertaking, not just monetarily, but also in the form of a true commitment towards advancing science. We have had several FDA-approved drugs that ended up being received by the patients at least 4 years before the approval actually happened, because patients had the trust in our team to get enrolled on clinical trials. So currently, we have several CAR T-cell clinical trials. Of course, CAR T's CD19 trial is also enrolling, but I'm most pumped about the CD19-22 study. And at this point, we have enrolled a handful of patients, and the initial response is very promising. Like I alluded to, the 5th patient who's on the clinical study currently in the hospital is really angry with me because I'm keeping her in the hospital right now. And she's completely fit because she has no side effects. So I think the future of CAR T-cell therapy remains promising, and at least the aim of our team is to cure 100% of patients who walk into our clinic. This is my group. These are a total of 5 clinical lymphoma MDs, but behind them is really a village that supports this effort. And thank you for really supporting our endeavor to be able to cure patients where previously therapies like this were unheard of. With that, I would like to introduce, and I really am honored to introduce Larry Firestein, my patient. I have taken care of Larry for several years, for 12 years, and I would love for Larry to share his journey with lymphoma.
Yes, I remember Dr. Kamdar right before senior prom, and kind of been through her life in terms of courtship, marriage, giving birth, become a father, grandfather, advisor, and I'm still with her. So what I'd like to do, because I don't have slides, and I'm just going to give you a Reader's Digest version of the last 15 years of my life. Starting in New York, New Jersey, where on my 60th birthday, which was 15 years ago this month, I just went for a physical. Everybody at 60 does. And my doctor, internist, who had been with me forever, said, "I'd like you to go see a hematologist." "Why?" "I just want you to." Okay, gave me the name of a hematologist, oncologist at Valley Hospital in Ridgewood, New Jersey. He kind of reminded me of Paul Simon in his appearance, but if you've ever watched the Seinfeld episode, he was a low talker. So it was very hard when he was telling me all these things and asked me to go get some tests and that we would talk later. So a few days later, a month later, calls me in his office and says, "You have mantle cell lymphoma." I said, "What? You have mantle cell lymphoma?" I go, I've heard of Lou Gehrig's disease because I'm a Yankee fan, but mantle, is that like Mickey Mantle has this? And he said, "No, it's called mantle cell lymphoma. I don't know what you're talking about." He said, "We're going to put you through some chemo, hyper CVAD." I'm reading because back then I didn't remember a thing he told me. We're going to get you right in, get your life in order, and be in the hospital. So I'm going out on a date tonight, and I'm going to a concert of Crosby, Stills, and Nash, so the hospital has to wait. He goes, no, that's not what's going to happen. We're going to get you in there, and 8 months later, and he drew me a picture, which I've saved since 2008, of what he was talking about, because he made this out. And 8 months of chemo, came in and said, you're in remission, it's gone. I went, wow, I don't have any hair, and everything tastes like metal, but it's gone. Then we fast forward, and I pick up from New Jersey, move back out here to Colorado, and start with a Dr. Mint, who was with the university, and then was suggested that I deal with Dr. Kamdar. I said, great. And Dr. Kamdar always gave me the lecture that it may be gone now, but we always have to have a plan B. Fine. And sure enough, in 2012, it came back. And now we're not going to do chemo. We're going to do ibrutinib, right? Ibrutinib, just want to make sure I pronounced it right. Seemed to be going on a good path. Scans were getting better. Tests were getting better. Counts were getting better. And then Dr. Kamdar came about a year later and said, looks pretty good, but it could come back. But right now, it looks pretty good. We want you in for blood work. We want you. And so I became a regular here every 6 weeks, to where I knew everybody. And they're all, if you're going to have a good hospital experience, this is the place to do it, no matter what department you go to. So finally, it's now 2020. We all talked about that pandemic, and at the same time the pandemic hits, it's back. But now we're going to try something new called CAR-T. And now, just like the first oncologist gave me, Dr. Kamdar gave me a chart, which I'm sure anybody like me would perfectly understand, take it home and study it, and then told me what she was going to do and how we were going to harvest. In fact, I took a picture, what's the machine called that harvests? Yeah, that. And I put that on my phone just so I could make sure what she told me was accurate. And understand, if Dr. Kamdar told me to just go off a cliff, I'm off. Whatever it takes. You're the boss. I love you to death. Just tell me what to do. Well, then it came time for the CAR-T. And what a great team of people. Erin and her team, unbelievable. And of course, they give you a book of CAR-T therapy patient guidebook. And if you've ever watched commercials on TV about a certain drug to take, that you might find this, and if you have this, don't take it if you've done this. I have them all. So when the CAR-T started, it seemed simple enough. You're going to harvest my cells, we're going to put them back in, and check into the hospital during the pandemic. But I didn't know that I was going to get everything that's in this book. Everything. There were no exceptions. I even checked them off and highlighted them. Neurologic toxicity, confusion, seeing, feeling, hearing, or tasting something that isn't real, severe headaches. They had to put me in restraints so that I couldn't even turn the TV on or off. Apparently, there were 2 days in this process I don't remember. They told me I've had spinal taps, that I've had all kinds of meds, that I've been in and out of scans. Don't remember a thing except that every morning they asked me who the President of the United States was, and what time it was, and what hospital you were in. And it seemed to get better each time. Writing became a problem, but I had everything in this book. That's why I saved it for future people that want to know what you can go through. I did. We go fast forward. It's now last month, 2023. Everything looks good. We still get the but right now I'm feeling pretty good. I'm working out. Everything is fine. You're fine. You have a beautiful baby that I'm about to meet and a husband who I've been dying to meet for years. And everything's okay except, let me go to the back page, one more term. The low immunoglobulin level, which has been a constant. We're striving for me to hit the number of 500, is that correct? And I've been hovering. So I've been trying to negotiate my way out of infusions. But it doesn't seem to play well with Dr. Kamdar. Even if I'm at 498, you're not at 500. So this has been something, and she said this will probably be this way for the rest of my life. And I'm willing to go for the infusions because they take good care of me. They give me ginger ale and Oreos. What could be better in the morning? Right? But just to tell you, it's been a long 15 years, but with what I've been through here and the people here, especially this young lady, I don't know where I'd be today, except eternally grateful. And if you'd like a copy of her chart, just see me at the end.
Anybody have any questions?
Yes, this is all pretty remarkable stuff and pretty impressive. But it looks like you've done, what, 20 patients or something along that line? And you had this slide up there about economic toxicity, and you're talking about hundreds of people for each treatment. Is there a pathway to make this commercially viable and economical so that it could be something that could actually be more than one person here and one person there? And how do you get there?
I think you're asking great questions. We have so far taken 195 patients to CAR T-cell therapy over the last 4.5 years. But the biggest piece of, I think, a conundrum that I don't think has been solved yet is the economic toxicity. The construct is $500,000, so whether it's insurance companies that authorize it or it falls on the payers, it is toxicity on the system at least. And that's why it's extremely crucial to develop products that are cheaper, and I think that's also one of our main aims here at the Gates Biomanufacturing facility.
It sounded like you said that you still have some space open for additional patients in the clinical trial. That's correct?
Yes. That is correct.
And how would patients or prospective patients contact the university to see if they are eligible?
So I think that's again a great question in terms of how do they actually come to us. And usually I will say that in terms of our network that we have built over the last, I started the program in 2014. Since then, I remember actually driving to New Mexico, driving to Wyoming, and along the state lines to make sure that providers were aware of our presence. So we have established connections with community providers who also see lymphoma patients. Usually the referrals come from them. And typically, we also have a very robust network within Colorado in terms of our primary care physicians who will identify the lymphoma patient. If there's a suspicious biopsy on hand, the first thing they do is contact us. In addition to that, we also have several ties with not-for-profit organizations like the Lymphoma Research Foundation as well as the Leukemia Lymphoma Society, where typically what a patient really has to do is talk to their hotline, and the hotline will direct that patient to the appropriate provider based on what or which specialist they're really trying to see.
That's great. Thank you so much.
In the pediatric space, there's also room on trial still. Luckily for us, we have less pediatric referrers because there's less centers treating pediatric patients. We maintain telehealth licensure in Montana, South Dakota, New Mexico, Wyoming, and Colorado to provide telehealth services when patients get referred to us to see if they're appropriate for our care. But we also have relationships with the 1 or 2 centers that treat pediatric patients with cancer in those states.
And I think one thing I did forget to mention is a government website called the clinicaltrials.gov. It will basically outline pretty much every single clinical trial for that subtype of disease that is available across the United States.
Other questions?
Is CU researching other types of cancers other than blood cancers for CAR T-cell therapies? I'm thinking of glioblastoma and the use of the polio vaccine for that kind of therapy. Is there anything going on here for other cancers?
I think there is a huge amount of research with CAR T-cell therapy in solid tumors. And I know Dr. Fry can speak to it, but in terms of on the adult side, my hero here is Dr. Jonathan Gutman, who's standing right in the alley over there. He's actually the director of immunology, immunotherapy, and he's actually directing a lot of these efforts towards making sure that CAR T cell therapy can also be delivered to solid tumor patients.
Yeah, I'm actually going to let Mike answer because the picture that he showed, the movie that he showed was from his lab, and he went through it quickly, but that was a solid tumor. And so Mike spends a lot of time trying to think about CARs for solid tumors. It's been more of a challenge for sure than lymphoma, but there's a lot of activity. But I'll let Mike take that one.
Yeah, I think as Terry was saying, our lab is kind of trying to really go beyond the blood cancers and use cellular therapy to target solid tumors. And part of the issue is that the tumors that we're targeting in children are bone and muscle tumors that are rare. But if you looked at a biopsy of a bone or a muscle or a brain, like with glioblastoma, as you asked, what you find is there's not a lot of T cells in there. T cells tend not to go into those tissues unless there's a reason to. And so what we're trying to do is understand whether the tumor is making chemoattractant molecules that make other cells migrate into the tumor, and they do in fact do this. And so we're kind of now making CAR T cells that will basically migrate into the tumor and then be able to kill the tumor and find the tumor a little bit better. And so that's kind of been our approach. Your question about the polio virus, I don't know of anybody in particular who's studying the polio virus in glioblastoma, but there's a fair amount of brain tumor work going on outside of CAR T cells. But I will say that even some of those people who have been studying other aspects of brain tumors have now really realized the power of CAR T cells and are starting to pick up, and we have a really promising CAR T program in brain tumors that is on our pipeline that will be coming to patients in the next probably 2 years. What do you think?
Mm-hmm.
18 months to 2 years is our goal.
Yeah, we're in the process of just beginning to start the early conversations about doing what we talked about tonight, which is to take something that came from a lab on this campus and turning that into a CAR T cell. It takes a while, but we're hopeful that in the next 18 to 24 months that we could get that into patients.
And maybe one thing that we didn't really touch on is why 18 months? Like, what the heck? Why is it so long? And the reason why it's so long is if we have a real promising target, everything is in the bag, it's perfect, right? Now we need to have a clinical grade virus to create the CAR T cells. And from today, we call up the various vendors who make these. They'll tell us it'll probably be about 15 months to get that virus produced in a way that we can then use it in the GMP facility. That's one of the major bottlenecks. And so another effort that we're really thinking about are alternative ways of introducing genes into these cells that are much more rapid. And I don't know, I'm sure everybody in the room has perhaps come across this term CRISPR, and it's a way that you can introduce or delete genes from cells. And so we're starting to think about that perhaps as a more rapid way of bringing these therapies without that, let's say, 15-month delay. Again, it's in the infancy right now, but that's kind of how we're starting to think about it. Exactly.
Yeah, remember the comment I made about if you Google CAR T cells, you come across using HIV to treat cancer. It's actually a modified version of the HIV virus, believe it or not.
And in fact, the headline that Terry is referencing was in the Wall Street Journal, and the title of it was HIV, the use of HIV to cure cancer, or something along those lines. But it was the kind of front page of the Wall Street Journal.
Other questions? I had a question. How does insurance decide what they're going to reimburse?
That's a tough one. So in the pediatric space, there's clear indications from the FDA of what Kymriah is indicated to do. So the initial trials only treated people that had had 2 relapses or more, or who had refractory disease. They only treated patients up to the age of 25. So 26 is not eligible for therapy, while 25 is. Strange but true. So there's very clear indications, and so insurance falls back on those indications and says, you're 26 years old, you're not eligible, we won't pay for you. Of course, another concern is people who aren't necessarily fully insured because of their immigration status, and so those people tend to fall into the holes of care.
I think maybe just to add to what Amy says, you know, every FDA-approved drug is approved for a particular indication, right? So if you come and have an ear infection, I could give you Augmentin for the ear infection, let's say. I don't think Augmentin is actually approved for ear infections, but no one's really going to be worried about it. I just read a prescription for Augmentin, you go, you pick it up. Nobody says, do you have pneumonia or an ear infection, right? You just pick it up at the pharmacy. These extremely expensive drugs, the insurance companies are laser focused on our use and using it outside of the indication. I think Manali can talk about, I don't know, is there a mantle cell drug? Or there is now, but at one point there were really narrow indications for adult non-Hodgkin's lymphoma that many patients weren't eligible. So the insurance companies are laser focused on making sure that we are not treating patients off-label. It pains us. We see, you know, I mean, I could speak as from the pediatric side. We see these kids who have relapsed once, and we know that they're not gonna, you just got to give them more chemotherapy and let them relapse a second time, and that's risking death. That's risking brain cells. It's torture for the patient and the family, but this is kind of where we are.
And patient number 7 actually turned 26 2 weeks ago, and so is no longer eligible for commercial care, which is why he's coming for our trial care. But what's the difference in those 2 weeks? Clearly nothing in his disease that he's had for several years, but that's something that the insurance can hang on and say, oh, not indicated.
But I think getting the payer blessing is so vital, because if we end up in single case agreement situation, then it's typically a 2-month delay because we need approval from insurance before we even collect the patient. I will say here, the leadership has been immensely helpful. Wells Messer-Smith in the audience has actually approved uninsured patients, has approved patients who have come from Mexico without, the ability to stay in the US, but they would die. So I will say that, it's something that we cannot sustain as an institution because it's economically toxic. But had it not been for such saviors, I don't think we could have saved these patients. So it's really important that the FDA approves it, but sometimes we get also stuck in the verbiage of the approval. For example, mantle cell lymphoma patients can get CAR T-cell therapy if they fail one line of treatment, and that one line of treatment has to be chemotherapy. I had a patient who went on a chemo-free pill, was in remission for 5 years, and then relapsed. I knew chemo wasn't going to work, but there was no way the insurance company was going to allow me to take him to CAR without giving him chemo. So I gave him chemo. It was unduly toxic. It did not cause the tumor to shrink, and finally got the approval for CAR T. So I think these nuances on the payer side is also really important that we just cannot ignore. I think our bane of existence right now to take somebody in a timely manner has been single case agreements. And single case agreements actually span through patients who are getting FDA approved products as well as CAR T-cell trials. So, especially for patients who get on Medicare, sometimes they believe that everything is going to be covered for, but it's not. If you don't have a supplement, there is no way you can get on any clinical trial in the United States of America. So these are just little things that I think people need to be savvy about when they go for insurance post-retirement. If they are on Medicare, they need to have the right plans to back them up if life throws a curveball.
I mean, I'll also just add, I think the first question was also about financial toxicity and how we can really do this in a sustainable way. And I think that, I think the statistic is that 33 cents of every dollar spent in this country, defense, finance, is on healthcare. 1/3 of every dollar is spent on healthcare. And now we've got these extraordinarily expensive medicines. The cost of Kymriah for children is about $500,000. For adults, it's $375,000, I think. There's other gene therapies that are going to come up to $3 million for a dose. How we as a country manage this is really something that is a huge challenge. And I think it's going to have to be a discussion between the providers and the insurance companies and the drug companies, but we're going to have to come to terms with how we do this. And I don't think any of us have a great answer for how to do this.
Obviously, an important topic of conversation for sure, and something that worth paying attention to. But I'm going to stop. I think we're at time, and I think we're going to. I want to thank the speakers, Manali, Amy, Mike, and especially Larry, for joining us tonight.